हाँ बस याद रखना, मेरे को बोलना ना पड़े क्योंकि इंटरफेरेंस हो जाएगा वीडियो। और एक चीज इधर कुछ और
बंद करना, बंद करना, बंद करना। जी सर, नमस्ते जी। This is Dr. Adhyar. So now we have our guest speaker, Dr. Himanshu Ojha sir, senior scientist in INMAS DRDO. Now I wish to request Mr. Siddharth, please introduce about sir and call him for the talk very soon. Over to you, Siddharth.
Yes, sir. On the behalf of organizing committee, we are very grand welcome to our chief guest and invite speaker Dr. Himanshu Ojha, scientist E, Department of CVRN, Institute of Nuclear Medicine and Allied Science, DRDO, Delhi. Without taking more time, I would like to invite Dr. Himanshu Ojha for his blessing and kind work about conference and some knowledgeable points.
Good afternoon to all the participant and learned speakers, and I really sincerely acknowledge the thanks for giving me the opportunity by the organizers that they given us a very wonderful opportunity to interact with the enlightened minds. I hope so. I'm able to convey. Hello.
Yeah, sir.
Yes, sir.
Audible,
Yes, sir.
audible, sir. No problem.
Thank you so much. So in this 45 minutes, I'm going to just talk about my research work. This is a research work under which we have investigated the potential of ethyl pyruvate as a promising candidate for mitigation and prophylactic application in radiation dermatitis. So I have a very small group which called CBN Protection Decontamination Research Group under division of CBRN in INMAS DRDO. So this is placed in Delhi. It's Delhi based. Hello. Hello.
Yes, sir.
Yes, sir.
Yeah.
आवाज आ रही है sir. आवाज आ रही है. Audible.
Okay. So what is radiation dermatitis? Because my topic is basically focused on radiation dermatitis. What is radiation dermatitis? So radiation dermatitis can be observed in victims, whether they've been exposed to radiation exposure such as in Chernobyl or in nuclear tech like Hiroshima, Nagasaki, or like sometimes it is experienced in the cancer patient who are undergoing the radiotherapy, as well as the occupational hazards. Like for example, those workers who are working in uranium mining, or they're doing some medical diagnostic. So such things happen during the occupational hazards also. So this is you can see that if I'm moving my cursor, you can see that this red portion, which is red in nature, and this blue portion is basically the necrosis of the skin. And ultimately, if it left medically unattended, it may lead to severe infection, sepsis, and ultimately contribute to the death of the victim. So that is why those radiation dermatitis required medical attention and urgent medical attention. So what is basically types of radiation dermatitis? So there are basically two types of radiation dermatitis. One is acute in nature. Acute radiation dermatitis observed in victims, those who has been exposed to very high radiation dose, that is a gamma, alpha rays, beta rays, or some transition metal ions. Those are radioactive in nature, and they have been exposed from few days to few weeks time, and that lead to first the red patches with desquamation, because desquamation is a condition of skin which is basically two types, dry or moist. So in that case, the moist and dry desquamations happen, and that lead to the blistering in the skin, in the affected part of the skin. This acute radiation dermatitis observed in the victims of, like, for example, Chernobyl, because the suddenly those workers who have been working in the nuclear plant, or the rescue firefighters who are there to for the firefighting purposes, they have been exposed to very high radiation gamma dose. And that is why this is the first time that in the history when this radiation dermatitis documented in the literature. The another type of the radiation dermatitis is called chronic radiation dermatitis. Radiation, for example, if it is of low dose and the victim is is exposing himself to the radiation for a long period of time, then it lead to a radiation dermatitis that is called chronic exposure, low dose radiation exposure for a prolonged period of time. This happened in case, like, for example, if you can somehow remember in 2010 in India, there was a very infamous Mayapuri incident happened because of some negligence, some procedural negligence. The a cobalt source has been passing to the junkyard, where some junkies have just break down that cobalt source, and they were not knowing that it is a radiation source, but they have themselves exposed to the low radiation exposure of the cobalt ray, the gamma rays, because that's coming out from the cobalt source, and they've been exposed for a long period of time, and that lead to the further radiation dermatitis in their on the back side of their body. That was the first documented radiation dermatitis case in India. That is available on the internet. So that is, if anybody is interested, I can go and read about the Mayapur. That is a very well documented case for the chronic radiation dermatitis. Now, what happened? How the cell responds during the radiation dermatitis? So you can see that in the normal cell, there is a epithelial layer, then there is some basal membranes, and then underline the basal membrane, the other body mechanisms take place, which help in taking the structuring, the microstructure of the epithelial and dermis layer of the skin. For example, there are the vasculatures that for the for the transportation of the essential component of the cell. They are macrophages to take care of the infection in the fibroblast cell, that basically the underlying bottom of the skin integrity. So all are there, but when they exposed to the high radiation dose for a short period of time, or a low radiation dose for a long period of time, then this phenomena, this stage is called, we scientists call it insult. It's called insult. And what happened in insult, that it lead to the deformation in the structural integrity of the skin, and they lead to the increase of reactive oxygen species and the activation of certain cytokines, like, for example, TGNF, TNF. So these kind of activations lead to the inflammation in the affected part of the skin. When this left unattended for quite period of time, then this injury response mechanism start taking place, because that if anything happened to our body have an intrinsic healing mechanism. So the body start doing it in the injury mechanism. How they do that? They first of all, because in order there certain cracks open up, certain structural integrity have been lost, so there may be a chance of getting infection. So therefore, the white blood cells, macrophages, they activated. In response to those, the macrophages, our cytokines mechanism activated, and then it lead to the, mushrooming up all those cytokines and macrophages at the site of the injury. And if our body is not able to, because in radiation dermatitis case, another important factor is radiation. Radiation also affect our immune system. So our immune system is, if got so it become imbalanced, so then our body is not able to heal up the injury part by itself. So it lead to the aberrant healing process of fibrosis. So it shows like a very delayed healing, the very excessive inflammation, and sometimes it lead to the infection in the affected part of the skin. So sometimes you require to amputate that part of the body if it is a hand or leg. So then you require to amputate that part of the body. So in order to prevent those kind of condition, we require the very good, the what you call therapeutic modalities to take care of the radiation dermatitis, so that they can quicken the process of healing as early as possible when you bring that thing into the place. Now, the choice of the cream is also depend upon the severity of the radiation dermatitis. So we normally, classify the radiation wound into 4 grades. The grade 1 is that mild atherma, or the dry desquamation with some hair loss. So here we normally apply Vaseline, and this is a very standard practice in the cancer patient. If doctors observe that there is a some mild atherma, they they just only advise to go just for the applying of some Vaseline, especially the Vaseline that is used for psoriasis. In the grade 2, sometimes, the patient's body mechanism is just like this. It elevated to grade 2, then in that the exposed parts he feels some pain, and the dry desquamation become moist in nature. So the moist desquamations is mean that your skin can be ruptured very easily. So here you require, the doctor start prescribing corticosteroid based cream. The other is that it become a confluent moist desquamation. Now the grade 3 becomes so much moist that even by touching the skin, it can be get ruptured. And if the situation becomes so pathetic, if it is elevated to grade 4, it lead to ulcer formation and necrosis of the layer, where infection can be happen. So for all the grade 1, 2, 3, 4, we required some medical attention. And it is that is why we require, because the moistness, whether it is a radiation wound, whether it is a excision wound, incision wound, in any kind of wound, the moistness play a very significant role. So therefore we required a medical topical formation, we required a medical, for the medical attention we require topical formulation that can cover the radiation wound. Third, it will maintain the moistness of the skin. And third, it will help in the cell adhesion and allow the anti-inflammatory nature of the drug should be like this. It should not disturb the inflammatory phase of the wound healing, but it should able to take care of the excessive inflammation if it happen in the wound. So that is why with this objective we have chosen ethyl pyruvate as a promising candidate. Why ethyl pyruvate? Because pyruvate is it's one of the compound which play a critical role in TCA cycle. So I don't think that the people require more explanation about what is TCA cycle. So this is an endogenous compound of our body. So pyruvate is what happened with the pyruvate. If I start using pyruvate, the main problem is its stability. If I take out that pyruvate and if I want to do some formulation with the pyruvate, then it become a problematic because it is not very stable. So what the scientists have done, they have developed a stable synthetic analog of pyruvate, is called ethyl pyruvate. People have tried many things: tertiary butyl, ethyl, sodium pyruvate, but but we have, but our group as well as the other groups in the, in England or the elsewhere, they have observed that ethyl pyruvate is the best synthetic analog of the pyruvate, and it has a very good anti-inflammatory and anti-apoptotic properties, and it inhibits the expression of nuclear factor kappa B, which is normally called NF kappa B, and it decreases the release of high mobility group box 1 in the chronic inflammatory condition, which is essentially required in radiation wound. Beside that, the other research group have investigated the hepatoprotective, renoprotective, neuroprotective, and cardioprotective effect of the ethyl pyruvate. So they have done this. They have also investigated the role of ethyl pyruvate in amelioration of organ dysfunctioning because of sepsis or acute lung damage or bone injuries, acute pancreatitis, strokes, and multiple sclerosis. So in our previous studies, beside that, we have also established the fact that the ethyl pyruvate is a very good reactive oxygen species scavenger. It reduced the systematic inflammation in the LPS mediated multi-organ damage. So by inhibiting the expression of cytokines and TNF alpha, and inhibit the methyl glycosol MGO induced cell apoptosis. So this is what we have established, and that is why it has become a very promising candidate. Let's try and see and investigate whether it will be able to take care of the radiation burden in the radiation dermatitis, both in the case of mitigation and the prophylactic. Now, what is the radiation mitigation basically? For example, if some person got exposed to the radiation, he became a victim. In that case, he required mitigation. For example, if I'm sending some rescue personnel to take out that person out of that exposed area, then I required the protection of that person. So in that case, I required a prophylactic application of that drug. So that is why it is called radio mitigative. So I'm sending some firefighters, I'm sending some police personnel, or I'm sending some medical personnel, then I required that these persons should cover the exposed part with that cream. And after having that, they should have some the personal protective equipment, and then they can go into the exposed part, which is called the hot zone or red zone. Fine. So that for the two purposes, I have done the radio mitigative as well as the radio protective potential application of ethyl pyruvate in radiation dermatitis. So this is the very small experimental design. I'm just keep it very simple, that if for the group of the rats, I have exposed them each rat for 7 days. The cumulative dose was 45 grays. After that, with the days, they start on the dorsal skin, they starting the formation of radiation scar, therma, ultimately moist desquamation, and then the radiation dermatitis. So here we start applying the hydrogel or the vehicle, for example, without the ethyl pyruvate, it's called vehicle. Otherwise, the hydrogel with the ethyl pyruvate is called hydrogel. So we start applying on that thing up to the 42 days. Then with the different time point and in different stages, we take out the samples. So we normally keep the physical observation. That is the one aspect of the studies, in order to understand the how one have been contracted, what is the score damage, and how the skin integrity can be measured in terms of thickness. Then we take out the skin samples, and we perform after the preparation of homogeneous, we undertook the biochemical assays to understand what is the protein content, how much the protein have been degraded, and what is the intrinsic antioxidant machineries response vis-a-vis to the ethyl pyruvate. And ultimately, what I presume that what we see, we only just come to accept that fact, that what we can see. So in order to verify it, we have performed the histopathology. So in the physical observation part, or we call the period of reepithelialization, we normally distribute the studies on the 4 time points. After 7 days, then 14 days, 35 days, and 42 days. And this period is not a random period. This is what we have, we have designed on the basis of the number of preliminary experiments. So if you see in a single time point, if I can make you understand that in the control groups or in the placebo groups, the wound contraction is not very significant. And we also undertook a standard compound, for example, Vaseline. So here we can see that it able to contract the radiation wound, the but the skin part is not covered with the hair, with the the hair. Then 0.2% ethyl pyruvate, 0.5% ethyl pyruvate, the healing is not sufficient. So you required at least 2% ethyl pyruvate to exactly to take care of the contraction part. But the contraction is not 100%. So in order to achieve the 100% contraction, we have to wait until 35 days. Here at least we can achieve, see 90% of contraction. But in order to bring that thing completely to the normal level, we require at least 42 days. And this what we observed in more than 3 experiments. So the 42 days is actually required in case if somebody got exposed to high radiation in a cumulative dose wise for a 7 days period time. At least we require 42 days to completely heal up. Then we have taken up we take the inner skin tissues, and we prepared the homogenates, and okay, that is another thing. So statistically, like this is the days wise and the sample wise, what we observed that the wound contraction was achieved in 42 days. Then this is the skin thickness is very important, because contraction does, because our healing mechanism and the rat healing mechanism is totally different. In the rats and the mice, the healing mechanism is that like it covers the affected part from the surroundings. It's basically contraction. But in our part, the healing mechanism is basically granular in nature. So in the affected part, the islands are prepared of the healed tissues, and then they start connecting with each other as the connective tissues or the collagen formation. So in that case, if we wish to correlate the healing process in rat with the human beings, we require to undertake what is the measure of thickness in the healed part. In exactly the thickness of the epidermis and dermis layer, what we achieve the good amount in 2% ethyl pyruvate after 42 days time. So that is why 2% ethyl pyruvate is the it's the ideal situation. It's the ideal formulation to undertake the for further study. Then we perform the biochemical assays. What we have because skin is all about proteins. So what is the protein content? So protein content was found to be very high in 2% ethyl pyruvate case. Then another parameter which is called melandihydes. Melandihyde basically prepared in those protein sample which has been disturbed because of some pathological situations. For example, if red proteins being exposed to radiation exposure, then there is a formation of melandihydes. So what we have observed that in the vis-a-vis to control samples, melandihyde formation is very less in case of 2% ethyl pyruvate. Very less in 2% ethyl pyruvate. So it means that both the studies protein content melandihydes are in sync with each other. Then we have also hydroxyproline is and the collagen formation is a very important marker, because ultimately whether the skin has been healed or not, it is all totally depend upon the collagen formation. So collagen formation assay, if we the data suggested that in the case of 2% ethyl pyruvate, what we achieve the highest highest collagen formation vis-a-vis to control, and it has not, and when we compared with the probability, it been found that it is what we achieve is it reached to its saturation level. So it is not possible if we go for 3% or 4%. We will achieve only to that level of collagen formation. So collagen formation data suggested that the 2% ethyl pyruvate is the best out of all the formulations that can be used for for treatment of radiation dermatitis. I don't know how it's uh, the catalase. Now the intrinsic antioxidant machinery is very important, as we know that in case of radiation exposure, whether it's a high exposure or low exposure, reactive oxygen species start taking place in our body. So in order to take care of that reactive oxygen species, our body have an intrinsic antioxidant machineries in form of catalase and amino acids and other type of enzymes that can take care of reactive oxygen species. And if that balance between the ROS and the antioxidant got tilted in favor of the free radical, then it is called oxidative stress. In case of oxidative stress, we require an additional exogenous compound to take care of that particular situation. So in that case we are using the ethyl pyruvate. But we have measured also we have measured that whether the applying this ethyl pyruvate formulation is able to take care of the intrinsic antioxidant message, and to our very separate, because we are not giving this ethyl pyruvate through oral route. We are just applying dermally. To our surprise, catalase and SODs, enzymatic activities, are found to be higher in the 2% ethyl pyruvate. I don't know why, because it's a basically a another topic of investigation for us, and definitely we will like to undertake more studies to understand that how if ethyl pyruvate formulation applying dermally is able to enhance the intrinsic antioxidant machinery. So that is another source of investigation for us. Then we have undertaken the heel tissue, the histology of the heel tissues, and what we have taken that in control sample. Control sample is one that is not being only radiated with the radiation. So what we can see large vacuoles here in the skin part, and these hair follicles are very completely lost. Then we compared with the standard and the vehicle, and what we observed that yes, that the healing took place, but yes, still the large vacuole part is there. When you start applying the 1% and 2% ethyl pyruvate exactly, then you can say that we are able to we can restore the skin integrity of the affected part of the skin of the rat. So and we have also measured the inflammatory cell in the affected part, and what we have we observed that in the 1% 2% ethyl pyruvate, we observed very less number of inflammatory cells. So it means that the process is not delayed. It is quicking the healing process. It is not being delayed, and it is being not a chronic radiation dermatitis wound, which is typically a problem. So ethyl pyruvate is working in the condition of radiation mitigation, because radiation mitigation is very difficult task. Stage wise, we have also checked the formulation on the stage wise, like after 24 hours of radiation treatment, what happened when the erythema onset, what happened when the dry desquamation take place, moist desquamation take place, and what happened when you compare the 2% ethyl pyruvate dry squamation and with moist desquamation. So it is very important time wise as well as stage wise. What we observed that the protein content wine or hydroxyproline or collagen data, all data suggested one thing, that 2% ethyl formulation is working on every kind of stage, especially the stage with the moist desquamation with ethyl pyruvate, because ethyl pyruvate is working as for the anti-inflammatory and anti-apoptotic wise, as well as the hydrogel part is contributing in the moisture, the is restoring of the moisture part, because it able to take care of the moistness of the skin. Because of that, that thing, they able to take care of the ECM deposition, they able to take care of the collagen formation, they able to take care of the restoration of the skin integrity. The biochemical assay wise, we have also observed the same thing, whether it is a carbonyl formation or a malonaldehyde formation or the thiol formation, on all the aspect which can indicate the protein damage part in the skin because of the radiation. The 2% ethyl parbate formulation with the moist tissue formation at that stage, which is a very critical stage, this formulation works very well and they can take care of that the, the healing part of the radiation dermatitis. So likewise, we also undertook the histology part, and you can see that we got a very good structural. There's no large vacuole, so it basically shows that the structural integrity is there. In the radioprotective part, just we had one more we had one more part of the step, that before the radiation we start applying the radiation on the part which is going to be affected by the radiation exposure, and that we have applied only for 3 days before the radiation exposure. So after the 3-day application, we put the radiation on the radiation dose from 7 days, 1, 2, 3, 4, 5, 6, 7 days, and cumulative dose of 45 days. Then after that, we start applying the hydrogel oblique vehicle, and then we perform the same studies. So for the sake of the simplification, we can see that the 2% ethyl pyruvate is able to take care in the part as vis-a-vis to standard. Standard is Vaseline in that case. So vis-a-vis to standard, the ethyl pyruvate is take care of that part. We have also seen the wound contraction part when we compared with the standard in the 2% ethyl pyruvate formulation. The wound contraction is far better as compared to standard, and the skin damage scores also suggest the same thing. The hydroxyproline and the collagen formation also found to be the higher in case of 2% ethyl pyruvate vis-a-vis to standard as well as the control. And again, we have also undertake the SOD and the catalyst activity part, and what we observed that yes, the affected part, they are the intrinsic antioxidant machinery is perfectly well and taking care of the reactive oxygen species if happen to be there, they are taking especially by the SODs and catalysts. So this is the you can see that the large vacuole parts in the standards, but here you can see that exactly the epidermis and dermis part with the collagen formation network here there. And the thickness wise, the 2% ethyl pyruvate is far better as compared to the standard. And there was the amount of inflammation, the inflammatory cells were found to be very less in 2% ethyl pyruvate vis-a-vis to standard like Vaseline. So in the conclusion part, if I have to summarize, I just want to stick to the time, and I hope so that last 10 minutes is available for the question answer round. So ethyl pyruvate is a hydrogel is significantly heal the radiation wound part in a very less time compared to standard, because standards takes almost 50 to 54 days. In our case, it is able to heal up in 35 to 42 days. So there is a gaining of 10 to 15 days vis-a-vis to the standard. So that is the beauty of this gel. Second thing is that we able to heal up the almost 100% damaged part, that 100% healing. We are achieving 100% healing vis-a-vis to Vaseline. Second thing is that it is not only gaining the time, it is also helping in the state wise. Third thing is that it able to comparatively lower the inflammation, and is able to make the collagen in a well arranged collagen fibers. Third thing is that we have found very less studies related to prophylactic. Yes, in case we are getting a very good result in prophylactic agent, we can use it for our rescue personnel also. Third thing, That these studies suggest that ethyl pivot hydrogel, which is have very good therapeutic as well as prophylactic effect, and therefore they can be taken forward for the effective quicken, fast healing, radiation dermatitis topical formulation. So this is a small groups of my students, and Miss Deepti, Miss Laspreet, they have contributed immensely toward the study particularly. And there are other students who belong to the chemistry, biophysics, because I have a very multidisciplinary group. They are these two guys is they have BIRAC project selected. They have been developing new microfluidic chips to detect that how the wound healing can be measured through as as, some sort of biosensors. So I always allow my students to think large and big, and no matter how we can contribute, because today I have just listening the first presentation, and we are just I have just read about the quotation that the world is only based on the three-person father, mother, and teachers. And we all are cover virtues from father and mother, but the third thing is a teacher. Teacher is play a very important role in the society, because if a teacher is allowed to show you the right path, then you can achieve whatever you can do right now. So I'm very grateful to my teachers also, Professor Rita Kakkar, and to my other teachers who have contributed immensely in me, and now the investment is now being gainful that I'm contributing further to my student. Thank you a lot. And if you have any question, I'm happy to take that questions.
Hello.
Thank you so much.
Yeah, ma'am.
Hello. Are you listening, please?
Yes, ma'am,
Yeah,
I
yeah.
can.
Yeah. Congratulations for such a wonderful and informative talk. Really so enriching to hear such eminent and distinguished personalities.
Yeah, ma'am.
My question for you, especially in pandemic, when this, all of the children, they were just, they were in the imprisonment. They were not allowed to go out also, and in, hot places where the temperature is so much outside. They were continuously there with air conditions being used. So when children, they are having such a delicate skin, and when they are exposed to solar radiation, so what is the effect on their skin and also on their immunity system?
Uh, Actually, there's some voice problem there, but if I can gather your question correctly, your question is that because of in Corona pandemic, people are not going out, and if they are going out in because of some reason, does it will affect their skin burn? I think that is your
Yeah,
question
right.
regarding the skin
Yeah,
burns and the inflammation part, and
yeah,
that is why you're
absolutely
trying to
true.
correlate. Yeah, okay, fine. Ma'am, actually, skin is the first barrier of our life defense. And what I personally feel that whenever you are going out, you should moisten your skin well, because that oily layer on the skin is helping in protecting any kind of, whether it's the dust, whether it's pollution, whether it's the sun. So that is you require to first moisten your skin, whatever you have, Vaseline or anything, because I personally feel Vaseline is best. Vaseline is best because not only be my personal observations, but it also in my other R&D aspect. I have what I have felt that Vaseline is best. If you keep on applying Vaseline, whether it is summer or it's a winter or it's a some rainy day, your skin your skin get perfect protection out of any kind of allergen, pollutant, or the other pathological conditions there. So in my terms, you require you should have a good sleeping pattern, you should moist your skins and eat healthy. And other
Right.
aspect is like reactive oxygen species, because the reactive oxygen species one who play a critical role in aging and wrinkles. So if you eat healthy foods with lots of antioxidant, then it take care of your the ROS part also. So that help you to build up your immunity. So I don't have any concrete answer in that regard. It's all depend upon the personal experiences and the the knowledge And in my case, because I have also worked on antioxidant, but I feel that antioxidant, having the antioxidant rich diet and good sleep with proper taking care of skin is